So, hello everyone, welcome to lecture 4 on animal kingdom. So, in last lecture, we learned about different phyla, like for example, we learn about coelentrates, we learn about cnidarians. In today's lecture, we will learn about different phyla, which are also a part of animal kingdom. So, this is the diagram which I always say that we need to remember always by heart in order to understand the classification of different phyla, different organisms into different phyla under the broad classification Animalia. So we know that under the kingdom Animalia, which are multicellular organisms, there are different organisms that are present on the basis of their level of organization. So on the basis of level of organization, we have cellular level of organization and tissue organ or organ system level of organization. So we have already learned about the poriferans that have the cellular level of organization. Now we are heading towards the organisms that have tissue organ or organ system level of organization. So in tissue organ or organ system level of organization, the organisms are further classified based on their symmetry, either as radial or bilateral. So in radial symmetry, we understood in the last lecture as well that any axis that passes through the center of the organism divides the organism into two equal halves. So this is about the radial symmetry. While in bilateral symmetry, so if I talk about the bilateral symmetry, in this case, the organism can be divided only into the two equal halves by a single plane. So only a single plane can divide the organism into two equal halves. So that is the bilateral symmetry. So in today's lecture, we will learn about the organisms that have bilateral symmetry, that are acoelomates or that belongs to the category of pseudocoelomates. So pseudo means false. Pseudo word is always related to the false. So pseudo means false. So that is they have false cavity. That is, they do not have true coelom, just like the coelomates. So they are pseudocoelomates, and we will learn about the ones that are acoelomates. That is, a word here represents the lack of anything, right? So the lack of, since it is acoelomate, so this means the lack of coelom. So we will learn about the organisms that are acoelomates, that is the organisms that lack the coelom, that do not have coelom or body cavity, and we will learn about the organisms that have the false body cavity. So basically in today's lecture we will learn about the organisms that belong to the group Platyhelminthes, which are also known as flatworms. And we will learn about the SK helminthes, which are also known as roundworms. So today we will learn about two different phyla, which is Platyhelminthes and SK helminthes. Both of them belong to kingdom Animalia. They have organ system level of organization. They have bilateral symmetry. And Platyhelminthes lack the body cavity. They are the ones which do not have the body cavity. So they are without body cavity. So they are acoelomates. And the second one is the false coelom. With the false coelom, that is the pseudocoelomates, which are SK helminthes. So Platyhelminthes are also known as flatworms. The common name of them is flatworms. While for the SK helminthes, they are commonly known as the roundworms. So we will learn about these phyla in details in today's lecture. So these are the basic characteristics. So when we will start with the basic characteristics of either Platyhelminthes or SK helminthes, we will again and again will hear the term organ system level of organization, or if we are talking about the symmetry, then it's the bilateral symmetry in both of them. So flatworms that belongs to the phylum Platyhelminthes. So flatworms or the Platyhelminthes. So flatworms or the Platyhelminthes live in marine, fresh water. They live in marine water, or they may be present in fresh water, or they may be present in the damp terrestrial habitats that are wet. So flatworms, which are the common name is flatworms. Basically, commonly they are known as flatworms. That is the phylum Platyhelminthes. They live in they may be present in marine, freshwater, or damp terrestrial habitats. So the flatworms are not always free living species. So in addition to being a free living species, flatworms also include many parasitic species. So this is very important because we will learn about various diseases which are being caused in humans also due to the parasitic infection of Platyhelminthes. So flatworms there are some species that are free living, but they may also include species that are parasitic. That is, they may cause parasitic infection in humans or in other organisms. For example, liver fluke or tapeworms. So flatworms, why they are called as flatworms? Because they have thin bodies that are flattened dorsoventrally. So you can see the structure here. So you can clearly see that they have a very thin body and they have a flattened structure which has been flattened dorsoventrally. And if I talk about the symmetry also, you can see that this organism can be divided into two equal halves like this, only along this axis, right? So this will be the one half, this will be the other half. So only along this axis it can be divided, and therefore they have a bilateral symmetry, right? So we will again come to this diagram when we will learn about the other excretory system of flatworms. But for now, flatworms are so named because they have thin bodies. They are flattened dorsoventrally, that is between the dorsal and ventral surface. And the word Platyhelminthes means the flat worm, and hence the phylum Platyhelminthes. Now flatworms have a wide range in their size. So they may be nearly microscopic free living species, while there are some tapeworms which are long enough, which may be up to 20 meter long. So they may be microscopic, for which we need the microscope, which cannot be seen with the naked eyes, to up to meters long of tapeworm. So there is a huge variety of, huge diversity even in the system on the basis of their size. We know that flatworms have triploblastic development. So what do we know about the triploblastic development is that we know that in triploblastic development, during the development process, there is a development of three germ layers, that is ectoderm, mesoderm, and endoderm, right? So in triploblastic animals, the three germ layers are present: ectoderm, mesoderm, and endoderm. However, despite being a triploblastic animal, that is, despite having all the three layers, these organisms are acoelomates. So we know that in coelomates, the body cavity is being formed by the mesoderm, right? So this is ectoderm, this is endoderm, and this is mesoderm. So the body cavity is being formed by the mesoderm. But in these animals, these organisms lack the body cavity. These organisms do not have this body cavity despite having mesoderm. So it might sound confusing to some, but we need to remember that these organisms are triploblastic, that is, they have mesoderm, but they are acoelomate. The earlier organisms were diploblastic, so there was actually no concept of having the body cavity, because we know that body cavity is formed by the mesoderm. But in flatworms, though they have mesodermal layer, but they lack the body cavity, and hence they are acoelomates, that is, animals that lack the body cavity. So, and how their flat shape actually helps? Their flat shape places all their cells close to the water in the surrounding environment of their gut. So if they are parasitic or if they are present in water, this flat shape actually helps in keeping all their cells close to their surrounding environment. So since they have a flattened shape, this flattened shape allows most of the cells to be in contact with the surrounding environment. So this is how this shape is also helping them. And because of this proximity, what is happening is that the gas exchange and eliminating the nitrogenous waste, which is the ammonia, can occur by the diffusion across the body surface. So basically the respiration or the exchange of gases or the waste material may takes place by the process of diffusion easily across the body surface. So if they are present like this, if they are flattened, so most of the cells will be in contact with the environment, right? So this flattened shape allows most of the cells to be in contact with the environment, allowing the greater exchange of water, gas, or the elimination of the waste across the body surface by the process of diffusion. Most of the platforms have a gastrovascular cavity. So we already learned about what do we mean by gastrovascular cavity. So a cavity in which the digestion also takes place and in which the vascular system is also present. So most of the platforms have gastrovascular cavity with only one opening. Those platforms lack a circulatory system, so they do not have a well-defined circulatory system, as we will see in other more advanced phyla. They do not have a well-defined circulatory system. However, the fine branches of the gastrovascular cavity distribute food directly to the animal cell. So all the work of the digestion as well as of the circulation is done by the gastrovascular cavity, which is present in the platforms. So these platforms, although they lack the circulatory system or the complete digestive system, they lack, but the fine branches of the gastrovascular system or the cavity distribute the food directly to the animal cell. Now, very important thing in these platyhelminthes group is that they have excretory system, which is called as the protonephridia. So this is really important. This may be asked that protonephridia is the excretory system of platforms, right? So if I start from the poriferans, which is the first phylum which we study. So in that we learn about cells like choanocytes, right? Then in the next phylum, which is the cnidarians, we learn about cnidoblast cells, cnidocyte that has nematocyst. So these are the specialties of some phylum. So if we talk about the phylum platyhelminthes, they have protonephridia, the singular of which is protonephridium. Fine. So they have protonephridia. This is really important. This may be asked. So platforms have excretory system, which is called as the protonephridia, and which form a network of dead end tubules. So it is a network of tubules. So they have tubule-like structure, that is tube-like structure. So these are tubules, and they have a network of these tubules. These tubules are connected to the external opening, branch throughout the platform body, which lacks a coelom or body cavity. So we know that in the platform, since they are acoelomates, they do not have any coelom or body cavity. Therefore, all the space is being occupied by the branches of this protonephridia excretory system, which is connected to the external opening. So this is like the protonephridia is basically the excretory system of the platforms. So when I say cnidocytes, so when we learn about cnidocytes, we learn that they have this specialized structure called nematocyst. In this similar way, the single unit, if I talk about the single unit of protonephridia, so the single unit is called as the flame bulbs. So the cellular unit called flame bulbs cap the branches of each protonephridium. So this can be made more clear with the help of this diagram. You can see that this is the protonephridia, the bulb system of a planarian. So planarian is an example of platyhelminthes. We will learn about platyhelminthes using example of planarian. So basically, protonephridia are branching internal tubules that function mainly in osmoregulation. So what is osmoregulation? That is maintaining the osmolarity. Osmolarity. So what is osmolarity? That is maintaining the osmotic balance. Whatever the salt concentration, water concentration is there, So it helps in maintaining the osmotic pressure or osmotic balance inside the body. So basically, protonephridia are the branching internal tubules. You can see that these are the tubules of protonephridia which are present all over the body of the platyhelminthes or the planarians. So this is a zoomed structure of a tubule. So you can see that the opening is in the body wall, and at the terminal they have flame bulbs, of which I was talking about. So at the distal end or at the terminal end, cellular units called flame bulbs cap the branches of each protonephridium. So each single unit is known as the protonephridium. So each single unit is called the protonephridium. And the branches of these protonephridium are capped by the flame bulb. So the branches are capped by the flame bulbs and it opens into the body wall. So this is about the tubules of the protonephridium. So now if I zoomed in into the structure of a flame bulb, you can see that they have tubule cells which are present, and there is a nucleus of a cap cell. So these flame bulbs consist of a tubule cell and a cap cell. Each flame bulb has a tuft of cilia. We can clearly see that they have tuft of cilia here. So cilia is present, a group or a group of cilia is being present, projecting into the tubule. So this is the tubular part. This lower one is the tubular part. This is the tubular part. Fine. So this cilia project into the tubules. And during filtration, what happens is that the beating of the cilia draws the water and solutes from the interstitial fluid through the flame bulb, releasing filtrate into the tubule network. So this is basically what happens is that whenever there is a beating of cilia, this beating of cilia allows the water to draw into the water and the solutes from the interstitial fluid. Interstitial fluid is the one surrounding these organs. So the interstitial solutes from the interstitial fluid through the flame bulb, releasing these filtrates into the tubular network, which is present here. And the moving cilia resemble a flickering flame. So when we see these moving cilia, it resembles like a flame of a candle, you can say, and hence the name flame bulb. So why this is called as flame bulb? Because the beating of the cilia resembles the flickering flame. So when you will understand the reason behind naming, you will remember it forever. So why these are called as flame bulb? Because when these cilia beats, they resemble to a flickering flame, and hence they are named as flame bulb. So the processed filtrate then moves outward through the tubules. So you can see that this processed filtrate then moves outward through the tubules. So it comes out through the tubules and empties as urine into the external environment. The urine excreted by freshwater flatworms has a low solute concentration, and hence, therefore, this helps in maintaining the osmotic balance, helping to maintain the osmotic uptake of water from the environment. Right? So this is how the protonephridia, which is the flame bulb system of a planarian, helps in maintaining the osmotic regulation or osmoregulation. So basically, what happens is that during the beating of the cilia, which is present in the flame bulb, it draws the water and solutes from the interstitial fluid. So interstitial fluid filters through the membrane where capsule and tubule cell interlock. So through this, you can see that the interstitial fluid is coming here, right? So the interstitial fluid is coming from the membrane, through the membrane where capsule and tubule cell interlock. And after this, it is released into the tubule network. So you can see the direction also. It is shown here in the blue arrows. So after coming from this membrane, it enters into the tubule network, and there the process filtrate then move outwards through the tubules. So this moves outward through the tubules and empties in the form of urine into the environment. Fine. So this is about the excretory system of planarians or platyhelminthes. Planarian is just an example. So protonephridia or the flame bulbs is important term. You need to remember this, that these are the excretory system. These are the part of excretory system of the platyhelminthes. So this is the general anatomy of planarian, which is an example of platyhelminthes. You can clearly see that it is a dorso-ventrally flattened structure, right? So it can be clearly seen in this figure. So they are flattened structures. They have flattened structures. And a planarian head has a pair of eye spots and lateral flaps that mainly detect the specific chemicals. So you can see here eye spots. And above the eye spots, they have ganglia. So at the anterior end of the worm, near the main source of the sensory input, a pair of ganglia, which is a dense cluster of nerve cells, is present that helps in the sensing of particular chemicals. The planarian nervous system is more complex and centralized than the nerve nets of the cnidarians. In cnidarians, we learn that they do not have well-defined nervous system, they just have nerve nets. While in planarian, the nervous system is little more complex and it is centralized. Experiments have also shown using the planarians, experimentalists have shown that these organisms modify their responses on the basis of their stimuli. Therefore, we can say that they have a well-defined, they have a more complex nervous system that responds to the particular stimuli. They show the responses to the stimuli. So these are different organs. We can start one by one with this. So firstly, we learn about the eye spots. So eye spots and the ganglia is present that helps in sensing the different chemicals or sources. Then they have ventral nerve cords. These ventral nerve cords from the ganglia, a pair of ventral nerve cords runs through the length of the body. So you can see here that these are the ganglia which is present. So the ganglia is present, and through this ganglia, a pair of ventral nerve cord, which is shown here in yellow, is being running throughout the body. Then they have pharynx. This is the pharynx. The mouth is at the tip of the muscular pharynx. You can see that at the tip they have a mouth. And digestive juices are spilled onto the prey. And the pharynx sucks small pieces of food into the gastrovascular cavity where digestion continues. So basically, firstly, they release the digestive juices. So digestive juices are released onto the prey. Upon releasing the digestive juices, the large chunk of food material turns into the small materials. These small materials are sucked by the organism and then it is digested into the gastrovascular cavity. Right? It is digested and supplied through the other cells to the gastrovascular cavity. So digestion is completed within the cells lining the gastrovascular cavity, which has many fine sub-branches that provide an extensive. So you can see these cells. So digestion occurs here in these cells, and the undigested waste is then ingested through the mouth. So it is ingested out of the body through the mouth. Now, planarians, if I talk about the planarians specifically, they can reproduce asexually through the fission. So they have excellent capacity of regeneration. So planarians have regeneration capability. So whenever you will learn about regeneration, you will always, most of the time, hear the term planarians. They have excellent regeneration capability. They can reproduce asexually through fission. The parent constricts roughly in the middle of its body, separating into head end and tail end. So basically, constriction happens here. So in the middle of the body, constriction happens. So one will be head end, one will be tail end, right? So these two ends are formed. One is head end and the other one is the tail end. So each end then regenerates the missing part. So the head end will finally develop the tail end also. The tail end will develop the head end. Therefore, the one organism will split into the two. So They have excellent regeneration capability. Whenever you will cut the planarian, it will generate the missing part. Apart from the asexual reproduction, sexual reproduction also occurs in the planarians. So planarians are hermaphrodites. And copulating mates typically cross-fertilize each other. So we will see what do I mean by this. So basically, by hermaphrodites, we know that they have both male and female organs within the same organism. Both male and female reproductive organ are present in the same organism, but they undergo cross-fertilization. That is, the sperm of one organ, will fertilize the ovum of the other organism. So the sperm of one organism will fertilize the ovum of the other organism. So it's a cross-fertilization. The sperm of one planarian will go and fertilize the sperm ovum of the other organism. The next example, so this is the reproductive system of the planarian. You can clearly see that in this figure. So this is the reproductive system of planarian. The pink color shows the female reproductive system. So this pink color is the female reproductive system. The blue color here highlights the male reproductive system. So as I mentioned that they are hermaphrodites, you can see that both male and female reproductive system are present in the same organism. So they have ovary as well as they have testes in the same organism. So both female and male are reproductive organs are present in same organism. Therefore, they are hermaphrodite. So these are hermaphrodites. That is, they have both male and female reproductive organs in the same organism. The other interesting part is about the cross-fertilization we see. So how is this cross-fertilization possible? This cross-fertilization is possible because during the mating, when these two planarians will mate, they will exchange the sperm. What will happen is that they will exchange so during mating, they exchange the sperm, and afterward, each planarian can produce eggs. So once the sperm is exchanged, they will produce the eggs. These eggs are being fertilized by the exchanged sperm. So this leads to the formation of fertilized egg. Right? So this fertilized egg is being formed by the cross-fertilization. That is, the sperm is of another organism and egg is of another organism, being a hermaphrodite. So this we need to remember that they are hermaphrodites. That is, both are present in same organism, but the cross-fertilization happens in these organisms. As I told about the nervous system also, so in nervous system, so basically, as I told about the nervous system, so they have a well-developed nervous system. So in this, they have brain. So they have brain, they have lateral nerve cord, and they have transverse nerves. So we see that the transverse nerves are run to all over the body. So they have a ladder-like nervous system, you can see. So they have a ladder-like pattern of the nervous system in which the lateral nerve cords run and connect one transverse nerve to the another transverse nerve. These are the transverse nerves, these two, and these are the lateral nerves, lateral nerve cord. Right? Oh, sorry. So these are the transverse nerves. Sorry, I'm really sorry. These are the transverse nerves and these are the lateral nerves. I'm really sorry. These are the transverse nerves that run like a ladder, steps of a ladder along the lateral nerve cord. So they have a well-defined nervous system, and this nervous system has although they have well-defined nervous system, but the nervous system is relatively simple. They possess a ladder-like structure of nerve cords called the nerve ladder. This is known as the nerve ladder. And this structure consists of two main longitudinal nerve cords which are connected by the transverse nerves, as I show in this figure. They have two main nerve cords which are being connected with the help of the transverse nerves. And the nervous system enables planarians to exhibit remarkable regeneration. So the regeneration property is exhibited with the help of the nervous system only, as even the small fragment parts of the nerve ladder can regenerate into the complete functional organ. So when it undergoes division, and the body divides into the head end and the tail end, the nervous system also develops itself properly into the complete nervous system. This nervous system in the platyhelminthes have excellent regeneration capability. This was about the planarians. The next example is the tapeworm. So the tapeworms are the second largest and diverse group of the parasitic rhabditophorans. So basically, they belong to the group rhabditophorans. The adult lives mostly inside the vertebrates, so they are parasitic. You will see that since they are parasitic, their structure is adapted in such a way that allows them to have parasitic modification. So they lack a mouth since they have to reside inside the body of any other organism, like for example humans. Therefore, they have to keep their body structure simple. In order to keep it simple, they lack some of the organs. And instead of that, they have some specialized structures. So instead of that, they have some specialized structures that we will see. Right? So these are the parasitic adaptations of these organisms because they have to reside inside the another organism, and therefore they have to be since they have to reside inside the another organism, they have to modify themselves according to their environment. So unlike planarians, these tapeworms lack a mouth and gastrovascular cavity. So they do not have mouth or gastrovascular cavity. They absorb nutrients released by the digestion in the host intestine. So they do not perform their own digestion. They just absorb the nutrients which are released by the digestion in the host intestine. Absorption occurs through the tapeworm's body surface, and basically you can see that this is the structure of a tapeworm. So in many tapeworms, the anterior end, so this is the anterior end, this is the posterior end. So this is the anterior end, which is the scolex. Anterior end, this is the posterior end. So in many tapeworms, the anterior end of the scolex is armed with the suckers. So they have suckers because obviously they do not have to perform the digestion. They only have to absorb the nutrients. And in order for that, they have suckers. And often they have hooks that the worm uses to attach itself to the intestine lining of the host. So if we see the zoomed structure of the scolex, you will see that they have hooks. These hooks helps in the attachment. So basically these hooks and suckers help the worm to attach itself in the intestinal lining of the host and absorb the nutrients from there. Posterior to the scolex is a long ribbon of units called proglottids. So in the posterior end, you will see that they have proglottids with reproductive structures. So which are little more than sex organs. After sexual reproduction, so basically they contain eggs and sperms, but after this, proglottids loaded with thousands of fertilized eggs are released from the posterior end of the tapeworm and leave the host body in feces. So basically, beside acting as a sac of sex organs, these proglottids are released outside. These proglottids get separated from the body of the tapeworm, and these are released into the fecal matter, thereby causing the fecal matter, which if being ingested or if comes in contact with another host, it then further can cause infection in that host. It contains fertilized eggs. So basically, if it is ingested, so you are ingesting the tapeworm. So it will get developed inside the host of the another body. So this is the life cycle of another tapeworm, which is a blood fluke, which is a trematode, another platyhelminthes. So example is Schistosoma mansoni. So you can see that the mature flukes live in the blood vessels of the human intestine. A female fluke fits into a groove running the length of the larger male's body as shown in the here. So basically the male body is large and the female fits into the groove of the male body only. So this is their adaptation in order to prevent the space which they require, right? So they live inside the intestine of the humans. So blood flukes reproduce sexually in the human host. The fertilized eggs exit the host in feces. If the human feces reach a pond or other source of water, the egg developed into the ciliated larva. And these larvae infect the snails. So snails here are the intermediate host. They are the intermediate host. Snails are not the primary host, but they are the intermediate host. Asexual reproduction within a snail results in another type of motile larva which escapes from the snail host. So this was a ciliated larva which is released into the environment through the fecal matter. And if it comes in the contact with snail, it causes infection in the snails. Like the snail acts as the intermediate host where this undergoes the asexual reproduction and resulting in the formation of a motile larva which escapes from the snail host. This larva then penetrates the skin and blood vessels of humans working in the fields that is irrigated with water contaminated with the fluke larva, ultimately causing the infection again into the intestine of the host. So this is the life cycle of a blood fluke, which is a trematode, which is again a platyhelminthes. Example is Schistosoma mansoni. The another is the Taenia solium, which I told here also. So this is the tapeworm. So Taenia solium, which is a tapeworm. The life cycle includes humans as a primary host and pig as a secondary host. So this is important. This might be asked that for the tapeworms, humans are the primary host, while pigs are the secondary host, right? The adult worm is modified for its parasitic way. It consists of a scolex, as I already told, and many proglottids, which become bags of eggs. So bladder worm attaches to human intestine where it matures into the tapeworm. So you can see that they have scolex and suckers. So as the tapeworm grows, proglottids mature. You can see the proglottids. So as the proglottids mature, they get eventually filled with the eggs. Eggs leave the primary host in the feces, which contaminate the water or vegetation. Livestock like pigs may ingest those eggs and they may act as a secondary host, as each larva becomes a bladder worm instead in the muscles. Now, when the meat, which is the rare or uncooked meat from the secondary host, is being eaten or ingested by the primary host, that is the humans, so if they contain the bladder worms, these again infect the humans. So therefore, it is very important that whenever we are eating the meat, it is very important that it should be cooked properly. Otherwise, there are chances that it might contain the larva of these worms that may infect the humans. This is the life cycle of the tapeworm, in which the primary host is humans while the secondary host are pigs. So you need to remember about Taenia solium, which is a tapeworm. We learn about Schistosoma mansoni, which is a trematode, which is again platyhelminthes, which is known as the blood fluke. And we learn about the planarians specifically in the platyhelminthes. The next group is the SK helminthes. So the body of members is circular in cross section and tapering at both ends. So this is like this. It is tapering at both ends, but it is circular in cross section. So when you will perform the cross section, it is circular in nature. Therefore, they are called as the round without segmentation. So they do not have any metamerism or segmentation. They are also called as roundworms. Nematodes are found everywhere. They are also known as nematodes or SK helminthes. They may be free living or they may be parasites in plants and animals, just like platyhelminthes. They have long cylindrical body with tapering ends and without segmentation. They are bilaterally symmetrical, so their body can be divided into two equal halves only through this plane. So this is, so suppose if this is the organism, its body can be divided into two equal halves through this line only. This will be the first plane, this will be the second plane. The alimentary canal is complete with a well-developed muscular pharynx. So they have a well-developed muscular pharynx. This is an important characteristic of SK helminthes, which is highlighted in NCERT also. So they have a complete alimentary canal with a well-developed muscular pharynx. All the three primary germ layers are present. That is, they are triploblastic, they have ectoderm, they have mesoderm, as well as they have endoderm. They have an organ system level of organization and have tubes within the tube body plan. So they have tubes act like body plan. The anterior end does not show a distinct head, therefore cephalization is absent. So cephalization word is used for the head. Cephalic, the word cephal is always related to the head. So cephalization is absent as the anterior end does not have any distinct head. So any distinct head is absent, therefore till this point you cannot see any head or brain-like structure. Cephalization is absent. Respiration occurs through the body surface by the process of diffusion. Circulatory system is absent. Excretory system is H-shaped. So they have an H-shaped excretory system. Consists of excretory canals, protonephridia, which we learn in platyhelminthes also, that removes the body waste from the body cavity through the excretory pores. And they develop from an embryonic renate cell. So this may be asked that the this is being formed from, in the embryonic stage, it is being formed from the renate cell. Excretory matter is ammonia, just like the platyhelminthes. It is similar to that of platyhelminthes. So these are some examples. So the left hand side example is of Ascaris. So this is of Ascaris, and this is of this is an encysted trichinella larva in the muscles. So in the muscles, you can see that it has formed a cyst, and so this is of trichinella larva, which is being encysted in the muscle. Fine. If you will see, you can clearly see the morphology here, which I was trying to make you understand there, that they have a cylindrical body. They have a cylindrical body. The body is cylindrical, round, but the ends are tapering, pointed. Tapering ends, right? The body is cylindrical and the ends are tapering. In the platyhelminthes, we see that the males are large and the female resides in the grooves of those males. While if we see in the Ascaris, we will see that in Ascaris, it's the female which is larger. This one is the female, which is larger than the male. The female is normally larger than the male in case of Ascaris. So what happens is that these Ascaris also cause infection. How? So eggs enter the body via uncooked vegetables, soiled fingers, or ingested fecal material and hatch in the intestine. So basically, the infective larval stage is being ingested along with the food, and these larvae then hatched into the intestine. The juveniles, which are the young ones, enter the veins. So they enter into the blood system. Basically, they enter the veins and lymphatic vessels, and through the veins and the lymphatic vessels, they reach the organs like heart and lungs. So basically, they reaches the duodenum first. So they reaches into the intestine. They hatches there. They penetrate into the blood vessels like veins and into the lymphatic vessels, and then they are good to go with the blood. So with the help of the blood, they migrate to the lungs, from where they are coughed up or swallowed again. So finally, Basically, from the lungs, the larvae travel up the trachea where they are swallowed and eventually reaches the intestine. So they may be covered up or they may be swallowed, and during the swelling process, they reaches the intestine. The larva again matures here, and the larva matures here and start having the parasitic behavior where it start having the food from the intestine. So these again, there the reproduction happens. The eggs are being released into the soil. These unfertilized eggs are released, which undergoes fertilization, forming the infective larva. These infective larvae are then ingested again with the food, and the cycle continues. So this is how the parasitic infection takes place. Now, this is the example of certain examples of SK helminthes. This may be asked because these SK helminthes are the ones that are associated with certain diseases. For example, Ascaris lumbricoid, which I told. So this is the ascariasis. It causes ascariasis, which is the intestinal infection. Trichuris trichura, which is a webworm. It causes trichuriasis, which is again the intestinal infection. Then we have enterobius, which is the pinworm that causes enterobiasis, which is again the intestinal infection. The most important ones are firstly the Ascaris lumbricoids. The next will be Wucheria bancrofti, which is the filarial worm. It is being spread with the help of the mosquito. So basically, with the help of the mosquitoes, it is a pathogen-borne infection. It spreads with the help of mosquito. So it causes lymphatic filariasis, which is the elephantiasis that leads to the swelling of the organs. The next is the Nicator americanus, which is a hookworm, causes the hookworm infection in the intestine. Then we have Encyclostoma duodenale, which is again the hookworm that causes again the intestinal infection. Then Strongyloides tricholis, which is the threadworm that causes again the strongyloidiasis, which is an intestinal infection. And then we have Trichinella spiralis, which is the trichina worm that causes the trichoniasis, which is a muscle tissue infection. So we see that these trichinella forms these cysts inside the muscles. So we see that structure also, that how the cyst is being formed inside the muscles. So it causes the muscle tissue infection. So Ascaris is one most very important example. The second is Wucheria bancrofti, which is lymphatic filariasis, that is elephantiasis. So what is so there are some few very rapid questions. What is the characteristic feature of excretory system in platyhelminthes? Do they have nephridia? Do they have Malpighian tubules? Do they have flame cells? Do they have antennial glands? So we know that they have flame cells or flame bulbs for the excretion. The next is, which of the following platyhelminthes is responsible for causing schistosomiasis? We know that schistosomiasis is caused due to the Schistosoma mansoni. And the correct answer will be Schistosoma mansoni. The next question is the digestive system of platyhelminthes. We know that in platyhelminthes there is no digestive or circulatory system. They have, instead of that, they have gastrovascular cavity, right? So the digestive system of platyhelminthes is absent. The next is, which of the following is a common nematode? So this is related to nematodes, that is the SK helminthes, that causes the lymphatic filariasis. That is, we know that lymphatic filariasis is also known as elephantiasis. So this lymphatic filariasis is caused due to the Wucheria bancrofti. The next question is the juvenile form, that is the young form of the nematode that causes lymphatic filariasis, is transmitted to humans with the help of, as I mentioned, that it is transmitted with the help of mosquito. So it is a mosquito-borne infection. The last question is, which of the following nematodes is responsible for causing trichoniosis in humans? It is, we know that it is caused with the due to Trichinella spiralis, which is the muscle infection, muscle tissue infection in humans. So this is a muscle tissue infection which is caused by Trichinella. So with this, we can end today's session on SK helminthes and platyhelminthes. In the next session, we will take up the another class. We will take up the another class of the phyla. So we will study about the another phyla. So for today, we'll end the session here. Thank you for attending.